negeri. Pancasila dalam artian Universitas Pancasila bukanlah sekedar tanda mengenal dan berbeda, melainkan memiliki kedalaman arti yang hakiki sesuai dengan misi dan tujuan. Memberikan peran positif dalam pengembangan serta pengembangan masyarakat berpancasila seperti yang dicita-citakan dalam pengubahan Undang-Undang Dasar 1945. Universitas Pancasila berniat dan bertekad untuk menghasilkan lulusan yang berjiwa Pancasila. Memiliki nilai dan sikap, pengetahuan, kecerdasan, keterampilan, serta kemampuan berkomunikasi dan mendukung pembangunan serta menjunjung tinggi nilai pendidikan. Sebagai wujud komitmen pengamalan dari nilai-nilai luhur Pancasila dan mengokohkan toleransi umat beragama, Universitas Pancasila membangun 6 rumah ibadah di tengah lingkungan kampus, yaitu Masjid At-Taqwa, Kelenteng Kebajikan Agung, Gereja Protestan Geraha Layanan Kristen, Gereja Katolik Santo Petrus, Pura Widya Santika, serta Vihara Dhamma Sasana. Dalam peran sertanya di bidang pembangunan kesehatan, Universitas Pancasila memiliki Fakultas Farmasi, dan hal ini merupakan wujud nyata seorang farmasis terhadap masyarakat. Sebagai fakultas farmasi swasta pertama dan terbesar di Indonesia, memiliki visi menjadi pelopor pendidikan tinggi farmasi yang unggul di tingkat nasional dan internasional, mengabdi kepada kepentingan bangsa dan kemanusiaan serta dijiwai Pancasila. Fakultas Farmasi Universitas Pancasila berkomitmen menjadi unggul dalam penyelenggaraan pendidikan, penelitian, dan pengabdian pada masyarakat untuk menghasilkan tenaga ahli dan profesional di bidang farmasi yang berkarakter mulia dan berwawasan global. Seiring berkembangnya dunia pendidikan, Fakultas Farmasi Universitas Pancasila pun terus berkembang dan memiliki 5 program studi, yaitu Diploma 3 Farmasi dengan akreditasi A, Sarjana Farmasi akreditasi unggul, Profesi Apoteker akreditasi A, Magister akreditasi unggul, dan Doktor Ilmu Farmasi akreditasi baik sekali. Dengan para alumni tersebar di seluruh penjuru tanah air hingga mancanegara. Hingga tahun 2022, Fakultas Farmasi telah mampu meluluskan tenaga ahli madya farmasi, sarjana farmasi, profesi apoteker, magister farmasi, dan doktor farmasi lebih dari 13.000 lulusan. Fakultas Farmasi Universitas Pancasila menerapkan model pembelajaran student-centered learning, problem-based learning, project-based learning, dan outcome-based learning yang mendukung proses pembelajaran terkini dan handal. Perkembangan model pembelajaran diterapkan untuk menghasilkan lulusan farmasi yang unggul dan bermutu. Dalam mendukung perkembangan dan kemajuan dunia farmasi, Fakultas Farmasi Universitas Pancasila telah ditopang fasilitas dan sarana prasarana yang lengkap, seperti halnya Laboratorium Teknologi Farmasi, Laboratorium Kimia Farmasi, Apotek Pendidikan, serta laboratorium-laboratorium lainnya. Fakultas Farmasi Universitas Pancasila dilengkapi dengan CBT Center dan OSCE Center untuk mendukung pengembangan kompetensi profesi apoteker. Fasilitas ini dibangun untuk mendukung sistem pembelajaran dan final exam lulusan farmasi yang berkompeten di dunia farmasi. Bidang kepustakaan, Fakultas Farmasi Universitas Pancasila memiliki perpustakaan yang telah berdiri sejak 1976 dan kemudian diperluas hingga diresmikannya gedung baru 2 lantai dengan luas lebih kurang 486 meter persegi yang diberi nama Perpustakaan Sunarto Prawiro Sujanto tepat pada bulan Juli tahun 2009. Dunia telah mengakui farmasi sebagai profesi yang besar dalam melakukan pelayanan kepada umat manusia. Seperti halnya Fakultas Farmasi Universitas Pancasila mempunyai tanggung jawab dan berkontribusi yang besar untuk menghasilkan lulusan yang kompeten di bidang farmasi. Dunia farmasi semakin tumbuh dari tangan lulusan terbaik yang telah tercetak. Tumbuh bersama Fakultas Farmasi Universitas Pancasila. Tumbuhkan terus prestasi untuk negeri Pancasilais.
Good morning, Dr. Juraitip. How are you?
Good morning. I'm fine. And you?
Nice to meet you.
Nice to meet you too. Very long time.
Yes. Yeah.
Thank
Okay,
you.
MC.
Yep.
Yeah. Please, MC Amalia.
All
Miss
right.
Amalia, sit.
Right. We're gonna continue to start our lecture for today. So to the LB audience, please stand by because we're gonna start now. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Shalom, Om Swastiastu, Namo Buddhaya, Salam Kebajikan, Salam Pancasila. Good morning and warm welcome from Indonesia. Our Honorable Dean of the Faculty of Pharmacy, University of Pancasila and FIC, and also our speaker, Dr. Ronald Juraitip, and also already joining with us, and also our Honorable Moderator for our international guest lecture today. And all participants in this guest lecture today who are very excited to discuss with us. It is truly an honor to have you all here today as we embark on an insightful journey of our knowledge and discussion. My name is Amalia Zahra, and I have the privilege of being your master of ceremony for today's session. We are gathered here to delve into medicinal plant tissue culture, an area of great significance and impact. We are fortunate to have with us an esteemed expert who will be sharing her valuable insight and expertise with us. And of course, today's discussion will be unlock new insight, gain valuable insight from an expert. Ladies and gentlemen, before we continue to the next agenda, we would like to invite you to sing the national anthem of Indonesia, Indonesia Raya, followed by hymn of Universitas Pancasila. For all participants, please be seated properly. Thank you so much. Before we proceed further, it's an honor to invite a distinguished figure who plays a pivotal role in guiding the mission of the Faculty of Pharmacy, University of Pancasila. Ladies and gentlemen, please join me welcoming Professor Dr. Apoteker Syamsuddin Magister Biomedik, the Dean of the Faculty of Pharmacy, University of Pancasila, to deliver an opening remark. To Professor Syamsuddin, the floor is yours.
Thank you, Ms. Amalia. Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Associate Professor Dr. Retnat Juraitip Bung Sintawekul as speaker, the Honorable Lecturer, student, and participants. As the Dean of the Faculty of Farmasi Universitas Pancasila, I highly appreciate the presence of Associate Professor Dr. Juraitip from the Department of Pharmacognosy and Pharmaceutical Botany, Faculty of Pharmacy, Prince of Songkhla, University of Thailand.
Enggak.
I'm sorry, Professor Syamsuddin, to interrupt you, but you on mute mode.
Oh, sorry. I got to know Associate Professor Dr. Juraitip during training grant and the specification and standardization of medicinal plant and related product on June 2015. The Department Pharmacognosy and Pharmaceutical Botany, Faculty of Pharmacy, Prince of Songkhla. She is a friendly and the master of the field. And topic presented today is medicinal plant tissue culture. The increasing demand for pharmaceutically important metabolites from plants has shifted the focus of research toward plant tissue culture. Micropropagation has become vital for the rapid multiplication of medicinal plant for the production of essential metabolites and pharmaceutically important compounds. I hope that through the discussion that emerged in this guest lecture, it can stimulate all students to be moved further in studying development in the upstream field of pharmaceuticals, namely the development of medicinal plant through tissue culture. Finally, I would like to convey my congratulations again to the audience for holding this guest lecture. Hopefully, many ideas can inspire the audience through today's guest lecture and become a blessing of knowledge for us to put into practice together. We hope can be discussion for collaboration, our research together from Dr. Juraitip. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Professor Syamsuddin. As we, Professor Syamsuddin, have a big hope for all of us to can discuss and know deeply about this medicinal plant tissue in culture. Okay, having set the stage for today's discussion, now we move forward to the core of our event for today. Medicinal plant tissue culture is one of the topics that particularly align with one of our major key missions, the development and advancement of natural products. As we continue to explore innovative approach in pharmaceutical sciences, medicinal plant tissue culture stands out as a crucial technique in ensuring the sustainable propagation, conservation, and enhancement of bioactive compounds to derived from the nature. And as pharmacy students, how can we involve to contribute the innovation of this field? It will be answered today. So to ensure a structured and engaging session, we are honored to have Mr. Pristiantoro, Magister Farmasi, with us today as today's moderator. So already joining with us. Good morning, Mr.
Morning, Amelia. Thank you.
Okay. All right. I believe all the audience is wait for you and our discussion for today. So without any further ado, the floor is yours.
Thank you. Okay, good morning, professor, student, and all participants. As introduced by the Master of Ceremonies, my name is Pristiantoro, and I, the moderator for the International Guest Lecture. In today's lecture, we will read about medicinal plant tissue culture. This lecture will be delivered by Associate Professor Juraiti Pungsita Wekul. Good morning, Prof.
Good morning.
Good morning. I hope you're healthy and full of energy. I am very excited for today.
I'm also excited.
Yes. Okay. First, allow me to introduce our speaker. CV, please. Yes. Prof. Juraiti Pungsita Wekul is an associate professor in Department of Pharmacognosy and Pharmaceutical Botany, Faculty of Pharmaceutical Science, branch of Songkhla University, Thailand. With over 2 decades of experience in pharmacy, she has made significant contribution to research in pharmacognosy, plant biochemistry, and biotechnology. Prof. Juraiti completed her bachelor degree in 1992 and from Songkhla of University, Thailand, and in PhD in pharmaceutical with second-class honor. She has that pursued her master degree in pharmacognosy at Chulalongkorn University, Bangkok in 1995. Her passion, the research, led her to Technik Universitas München, Germany. Her academic career began as lecturer in 1995 before being promoted to assistant professor in 2005 and eventually reaching the position of associate professor in 2011 until now. With a strong focus on pharmacognosy, plant biochemistry, and plant tissue culture, Prof. Juraiti has produced numerous scientific publications that contribute to the advance of the pharmaceutical science and biotechnology. One her main research focused in study on kratom, Mitragina speciosa, particularly in alkaloid biosynthesis, chemical analysis, pharmacological effect, and as impact of human health. Over the past 5 years, she has published numerous scientific articles. Metabolic engineering of yeast for production of kratom monoterpenoid alkaloid, published in Metabolic Engineering, 2024. Rapid metraginin quantification and fingerprinting of kratom product using XPTLC published in Phytochemical Analysis. Study of the effect of kratom extract on human gut microbiota, published in the journal Science of Food and Agriculture. Effect itraconazole on pharmacokinetic of mitraginin and healthy volunteer in AGS Pharmacology and Translation Science. A 4 years retrospective study kratom in discontinuous is an impact hospitalized patient, published in Journal of Health Science and Medicinal Research. Through her research, Prof. Juraiti has provided new insight into the potential benefit and risks of kratom in medicinal pharmaceutical field. Her expertise in pharmacognosy and plant biochemistry makes her one of the leading research contributing to the advance of natural product-based pharmaceutical. With a strong dedication to research and education, Prof. Juraiti continues to play an active role in developing innovative pharmaceutical solution while mentoring the next generation pharmaceutical science in Thailand and international academic community. All right. That has introduction of our speaker. We can say Prof. Juraitib in expert today the topic. Before the presentation begin, here are the instruction for this lecture. The lecture will be delivered for a maximum 45 minutes. After the lecture, Q&A session will be open 15 minutes. During Q&A session, participant can ask question directly for Juraitib. All right. Those instruction. Next, let the main agenda for this event. Prof. Juraitib, if you're ready, please begin your presentation.
Okay, I can share my slide. Can I share my slide?
Please, computer.
Yeah, I can share,
Yes.
right? Okay.
Okay, thank you.
Do you see my slide?
Yes, I see.
Okay.
I see, Prof.
Full screen. Yes. Okay. Now I'm ready. I can start.
Yes.
Okay.
Yes, Prof.
I start my speaking. Okay. Good morning, everyone.
Good morning.
I'm
Good morning.
very honored to get this opportunity from Professor Samsudin as a guest lecturer today. And the topic of the medicinal plant tissue culture, which interesting and also in my expertise, study of that. I use plant tissue culture for like a factory of the building of the secondary metabolites. So with my background, you can see that I'm interested in the biosynthesis of the medicinal plant. So if we would like to study the plant, we're looking in the intact plant in the nature. Of course, we have to build artificial one to study in terms of the gene, enzyme, and metabolites in the plant. So we need a control condition of the plant cell. So plant tissue culture is the one way to do. So today I have about 45 minutes to finish my about 80 slides. So I will a little bit fast in my lecture. I will focus as I outline here. That is, I have learning objectives that I would like to convey all information to you. Discovery of plant tissue culture, how what the history, why the plant tissue cultures began to be interesting, especially nowadays, because the plant tissue culture is like a media between the nature to the molecular biology. The culture media, how can we grow it, a composition, establishment, manipulation, and I will finally speak about application. So I just only three messages. That's a knowledge about basic knowledge about the history. The second is how to establish a plant tissue culture. The third one is how can we manipulate plant tissue culture. So we back to the basic knowledge. This is a comparative between animal cell, plant cell, and bacteria. A bacteria is quite simple structure. This is a membrane. They have unorganized the material in the cell. So bacteria is quite easy to grow, and compared to the complicated animal cell and plant cell, is a little more organelle inside. The organelles have a function, a specific function, and an animal cell and plant cell are also different in terms of the cell wall, chloroplast. So it means that when we focus to the cell, plant cell, we say that it is an autotrophic organism. What does it mean, autotrophic? Autotrophic, it means that they can grow by themselves. They have a factory, they have energy, and they can grow with the carbon dioxide, with the water. And so with the supplement of nutrient, they can spontaneously growing. So therefore, the plant cell is easier to grow than the animal cell, and then they're easy to manipulate. So many years ago, they also mentioned that animal cell, we need to grow it and we heterotrophic organism. That is the thing that's the difference between animal cell and plant cell. But the plant cell in nature, it is autotrophic. If we change a plant cell to the plant tissue culture, we move the plant cell from the autotrophic to heterotrophic organism. So it means that we have to supply some nutrients, some vitamins, some growth hormone to them, and then they can divide it and they can grow and they can differentiate the way that we are planned to do. The characteristic, this table compare between microbial and plant cell tissue culture. The microorganisms, they have a very intact membrane, and the plant cell, they have a cell wall, is a rigid structure. So comparative in terms of the size, the plant cell is bigger. Shear stress, because of the cell wall, it is very sensitive to the shear stress. Duplication times that of course take a little bit longer, not a little bit longer, this is sometimes months, 30 days, for example, but microorganism is less than 1 hour. This is a disadvantage to grow. We need times. Fermentation also reflect to related to duplication time. For the accumulation, this is very important. The structure of the plant cell, they have a spatial vacuole. So the vacuole, some kind of the organelles like a chloroplast tract secondary metabolite. So if we would like to study secondary metabolite, we have to open the cell. Unlike microorganisms, that's because of the structure so simple, they cannot accumulate the secondary metabolite. The media will be the place that microorganism or bacteria release the component out to the medium. So this is different from this table. One thing that's when we grow the plant cell, we need days to grow. A second is sensitive to shear stress. We cannot shake it very fast. We should not rigorously shaking, otherwise the plant cell will destroy because of the shear stress. So I come to the history. This is the thing. If we study for the plant, you can read in the textbook. This is I would focus. This is this region. You see, you disembarking of this tree, and then you see that the root put all media or food or nutrients to here. So the plants can grow in the young shoot, but without the food or nutrients, so in this part, the cell is swollen. So increase the volume. So Duhamel said that it is the evidence. There's a picture show some kind of the enhancement of the volume of the cell. They call callus, C-A-L-L-U-S, the callus. So by this discovery at the time of the Chuan and to discover the cell theory, they have the very good microscopic, and later on, the Gottlieb Haberland, the botanist, say that the plant cell can have totipotency. This is a technical term of the word that we talk about the plant tissue culture. They have a totipotency. They have a chance to grow. If we isolate the plant cell, they can increase the volume, and then they can divide the cell unlimitedly. But at that time, he said that totipotency, but he observed only one cell in some kind of the medium, but increased the volume, but undivided cell. He a little bit disappointed about that, but later on they found the substance that very essential to increase the number of the cell is the Wool hormone. So the Wool hormone is the most important in plant tissue culture. So the hormone, the Wool hormone, they find later on after Gottlieb Haberland launched a totipotency theory. They found Guthrie and White found the auxin utilization. This one, auxin, we know now, auxin is a kind of the hormones that they increase the cell, the division, the cell division, and also growing of the root. And another hormone that's very important is coconut milk. That is a kind of the cytokinin. This cytokinin is another type of the hormone that is very important for the jute production. The finding or discovery of the auxin and cytokinin comes to the principle of the micropropagation, the word that we can divide the plantlet unlimitedly by the working with the balancing between auxin and cytokinin. So the control of the organogenesis is the one that we can play on the balance between auxin and cytokinin in the plant tissue culture. So this is the first demonstration you see in early 19th century. Later on, about 50 years, 50 years, they just finished the totipotency all picture. So this show the Nicotiana tabacum. They isolate the single cells and looking in under a microscope. But of course, they grow this cell in the media with the Wool hormone, and then they found that the cell are divided into be a group of the cell. This is still like a group of the cell, undifferentiated cell. They call these is colors. From these colors, we they performed the ratio of the organogenesis between auxin and cytokinin, and they found that if they put a little bit more cytokinin, they have a lot of the jute. So we call this a micro jute, and from that jute we changed to another medium, and for the root production, and we have the full intact plant. From this intact plant, you can grow in nature. So this is kind of totipotency idea that is completely demonstrated in 60 years. So therefore, the plant tissue culture, they're not only to make produce the small plantlet. We now knowing that the decorated plants always produced by this way because it's cheaper, and then it is very fast, and we can stabilize the plant, in terms of the morphology. So from this idea, they summarize by this basic step. We can take some part of the plant, put in the media, and then produce the colors. From that colors, we produce a suspension, and from that suspension, you can go back to the colors or colors to suspension, whatever you want. And then you can produce the root, produce the jute by the ratio of the hormone, and you get the plantlet. So you have succeed that. So the keyword of the plant tissue culture, it is the undifferentiated cell. This is colors here, suspension here. You cannot see any type of the plant, let's say jute or root, unless you change the ratio of the hormone, then you can get it. The another word is in synthetic medium. So the medium that's have they put kind of the organic nutrients, inorganic nutrients, vitamin, and also Wool hormone inside, and then they can grow under aseptic condition. One thing, undifferentiated cell. Second is a medium that's produced synthetically and aseptic condition. So it means that we can control the growth of the plant tissue culture. We can control the factory of producing the secondary metabolite, then we can then manipulate the cell what we want. So the type of the plant tissue culture, there are many types. So I will skip this slide. I will show a picture. This is a colors culture. This is one plant that I initiated when I came back from Germany, and then I would like to produce easier color cell culture. This undifferentiated cells, and from these colors, I can produce the suspension and check it in a specific RPM to let them get the oxygen and a medium. And there's another type that I took the picture from the internet. That's a protoplast culture. This is you can isolate the cell and then remove the cell wall. You get the protoplast. From this protoplast, you can grow and you can, how to say, like hybridization between plant species. So this is very important in the, how to say, like a decorated plant, like a tulip, like many types of the flowering plant, they use this type of the hybridization. Another organic culture, for example, hairy root culture, also very interesting. I have got one from Gratom, hairy root culture, but unfortunately they cannot grow, they cannot produce any alkaloid, but a lot of triterpenoids they are produced. And embryo culture, that's the one thing that's like a crops like Hevia brasiliensis and rubber tea, or the palm tree also how to say succeed with the embryo culture. You can see there's a lot of type of the plant cell that we can work on it. We can produce a stem cell as well. A stem cell. What is a stem cell? Stem cell is unlimited growing. Maybe some part is a color cell, but color cell is not a stem cell. Stem cell is a color cell, but color cell is not stem cell. We have to find out which kind of the factors that colors have the stem cell. If the plant stem cell, they can produce the secondary metabolite. If you're growing a stem cell and you can, how to say, like sustain the production of the secondary metabolite. It's quite interesting area. You can looking in the textbook and study on the online textbook. This concept that from colors we can change the hormone and differentiate to be a plantlet. So it means that you can produce a colors, you can manipulate the gene inside, and from that gene, maybe they have a specific a characteristic, a specific morphology, like genes that produce a little bit more secondary metabolites, and then you grow to the plant, and plant have a little bit more yield of the secondary metabolite. This is the concept. So what kind of X size from the plant? So it means that which part of the plant cell is appropriate to growing as the cell culture. So X size from which part? For me, it doesn't matter. You have to find out which kind of tissue along the plant is a meristematic tissue. Meristematic tissue, they have a specific the characteristic that they can grow after, you say, like a division unlimitedly. You can use the shoot, you can use the leaf, but the leaf is not too old because of if it's all the cell is a little bit different. I mean, in terms of the division, the bud is quite active, and the root also active. Maybe in the here root tips, this is a bit more. So stem, a young stem is quite all right. So it doesn't matter. I tell my student, you can looking at the plant, and you can try which kind of the X plant. You can start with the plant tissue culture. Once you get it, it is enough. So you apply, after that, we can apply in many ways. But for the Faculty of Pharmacy, or my interesting, I'm thinking about study of biosynthesis of that compounds. Now I'm interesting in alkaloid biosynthesis. So that's why kratom is my interest right now, because of the part of the combination between the terpenoid part and shikimate part. That is, I would like to know how is it growing. You can study about the review of the updated paper of collaboration with Professor Michael Jensen from the Danish Technology University. Another thing is produce a novel compound. A novel compound is quite, of course, interesting, yes. And we can also like a conservation for some plant that's an endangered species, and continuous supply of the uniform of quality. Meaning that if you grow, you have a good plant and you make a tissue culture and you duplicate it, you have a uniform quality of the medicinal plant. So this one, right, application. So I just focus in the area that I'm interested in. This is a picture from my friends from Konkan University. She studied about the hairy culture of the Artemisia annua producing artemisinin. So she combined with the molecular biology by introducing the key enzyme here, the amorphidine synthase, together with the green fluorescent protein. And then she followed that the hairy culture that produced by the agrobacterium rhizogenes that have a gene introduced to the root. So it means that because of the amorphidine synthase incorporate to the hairy root culture, then this hairy root culture produce a higher yield of the artemisinin comparatively with the non-transformed amorphidine synthase. This is another study with kratom. I have also introduced the genes of the tryptophan decarboxylase, and then we found that the mitagenin is a higher amount than the normal root culture. So this is a combination with the molecular biology. So the micropropagation, this is another way that we can manipulate. This is show the ratio, the differential of the ratio of the auxin and cytokinin. So you can study that only auxin, they cannot grow the complete plant. Only the cytokinin, they also not grow a good plant. So the ratio of this depends on the plant species. You have to study about the ratio between auxin and cytokinin to get more plant healthy characteristic. Let's say 1 milligram per liter of the NAA and 0.1 BA in this case, for example. So this is another picture that I got from internet that is showing the suspension. So from suspension, if you remove all media and then you put only the cell, sterilization condition to the solid media, you get the colors. From the color, you change the hormone, you get the shoot, change hormone, you get the root, and you have an intact plant. This is concept of micropropagation. So that's in my interest at that time, and I would like to produce the colored culture a little bit colorful because currently Ternatia blue pea, this is a dark violet, dark blue, right? Color. I would like to see the colors culture and try to produce the colors culture, but at that time it was unsuccessful. I got only the undifferentiated cell, and then under the light they have a green. It's very green after grow for some for times, and then a little bit more intense green. But growing in the dark, there's nothing to growing of the chloroplast at all. So meaning that the light intensity also affected to the growing and production of secondary metabolite of the cell. So this table, I got it from Professor Singh, my former advisor. He wrote a very good review about this. This table, I would like to show the advantage of the plantage culture one thing, this is in case of ginsengicide, the culture must produce higher yield than the normal plant. Then it's, we say, benefit to use the tissue culture, benefit for the investment, everything about the tissue culture. So the culture must produce higher yield than the plant. Second, the plant tissue culture, not only a secondary metabolite, not only get from one plant, you can find another plant produce also the same compounds. So meaning, it doesn't mean that you would like to have a berberine. It doesn't mean that you only produce a thallic minor, but you can get it from Coptis japonica. So you can compare which plant tissue culture produce a little bit more because of these two plants is very close to each other. That is can produce berberine, isoquinoline alkaloid in the plants. So that's why you can looking for other, but kratom is a little bit different. They have friends of kratom, the mitagenin only produced in the Mitagena species, that is the thing. But another Mitagena species is also share some biosynthetic pathway about this. So we have to think about in comparatively. So Nicotiana tabacum, this is one plant tissue culture can produce more secondary metabolite. This is the advantage of using plant tissue culture. This depends on the method that you have in hands, like you have a specific advance of the detection, you have an advance of the separation, you have an advance of tasting activity or something like that. So you can give a little bit more importance to the tissue culture. So nowadays, the plant tissue culture not only thinking about the alternative source of the secondary metabolite production, but you can produce a plant tissue culture as a cell, producing how to say, you can use it for the cosmetic purpose. You can use it for now. You can key to the internet about the plant tissue culture and cosmetic. This is a lot of the cosmetic supply company using a plant tissue culture as a source of the secondary metabolite. They call some, you can look at in the internet, like a Switzerland company, Swiss company, and also Thai company also produce that. I mean, very famous Swiss company producing CBD, yes, from the cannabis sativa using a plant tissue culture. So this is unlimited actually. So the laboratory, this is our laboratory. We have a shell for the incubation. This is a plant that's we produces by the micropropagation. We have the colors here. We have a small shoot here. So now I produce a plant, I mean, uniform plant of kratom using a plant tissue culture. So this is the plant in the artificial soil. So this is the area that if you start, I don't know whether in your faculty have the plant tissue culture section. Please, the faculty have tissue culture laboratory. Do you have?
No, Prof.
No, you don't
Yes.
have. Okay, so if Professor Samsudin would like to establish the plant tissue culture laboratory, this is the area that I draw from our laboratory. We have preparation area. This is you have so many things to prepare, media, autoclave, everything, and you have an incubation area. You have a transfer area. This is quite simple, and it is not. I think animal cell culture scoring is harder than the plant tissue culture. This is easier, I think. Control temperature 25 degrees Celsius the whole 24 hours. So we have two air conditioners to control the temperature. So this is the laboratory. So what we need is laminar afrocarbonate. This is very important, but it doesn't matter it's horizontal or it's vertical because of the plant is okay. And when we have a vertical flow of that. So the HEPA filter, the UV light is necessary for sterilization. Incubation area, shaker, you need a shaker, yes. So you see our innovation, when we don't have enough space for the shaker, they have a double layer of the plant shaker. So with the rubber rings, so this is interesting. My student did that. So the aseptic technique is a little bit important in plantage culture technique. You have to select the right aseptic technique for appropriate for that step of the working. Autoclave is quite important to sterilize of the media, sterilize of the everything, water, braid, forceps, everything, the autoclave, right? And UV exposure, this is UV carbonate, UV exposure, so just like a surface clean. A membrane filtration, that's using when you would like to make a filter sterilization. You have a membrane 0.22 micron to remove all bacteria, and then a hot air oven. This is for the glassware. That's a long time. This is 3 hours, very high heat. You may need hot air oven, or you may need the autoclave, and then they dry in a hot air oven is okay. Chemical sterilizing agent like ethanol, this is 70% ethanol, and some bleaching to remove the surface bacterial of the plant, sodium hypochlorite. Bacteriostatic agent like antibiotics. This is like a penicillin, like a cephalosporin. You can use that. Number 5, number 6, they use to remove the bacteria. I mean, they remove from the plants that we initiate color cell. This is sodium hypochlorite. That's using in plantage culture, we call collox, but you don't need it. You can use Hyder as well. This is the carbon source like sugar, glucose to for the energy production. Mineral, micronutrients, macronutrients, vitamins, plant growth regulator, and some gas, oxygen, carbon dioxide. So, right. This is the name of the plantage culture media. We very familiar to the MS media, Molachike and Skoog. We familiar with the Gambot V5, and we maybe heard some Linsmeyer and Skoog LS media. And another that I use for the growth hormone initiation is woody plant media. So you can choose it, and I won't talk in detail about how to prepare it, but you can looking in this is phyto laboratory. They supply the complete set of the nutrients with the vitamins, so it's very easy to prepare the culture media. This is a little bit small, but I would like to focus in some part. This is I put the colors differently. You see, that is a group of the nutrients. The first one, you see that MS media, they contain a high amount. We call this macronutrient. And the violet one, this is very small, but we need it. Very small, is a micronutrient, right? And the carbon source is you can use glucose or sucrose. And another is pink color. This is the vitamins. This is a vitamin base, vitamin A. It's a mixture of vitamin. So MS media, white media, gamma B5 media, you can see they are different of the supplement of the nutrient, meaning that you have to find out which one is appropriate for plant culture initiation. Normally, the basal media is MS and B5. You can start at the beginning. We have tried with the MS and B5, but it's not successful for the plant for the kratom, because the kratom is a woody plant, it's not a herb. So I changed my mind to use the woody plants. If you're interested of that, you can looking for the composition of woody plant media, and you can compare with the two types, then you can see the difference between the micronutrients, macronutrients, and also vitamins are different. So you can see the biochemical difference of the media to the plant cell. So what, I'm sorry, here is Thai word, but the composition organic nutrients quite important. This is nitrogen, phosphorus, and sulfur supplement. You can see nitrogen, like nitrate, potassium nitrate, right? And phosphate, that's you see the type of the salt. In the plant culture medium, they put a salt inside that they can dissolve completely. Once you see the precipitate, it means that your media is already damaged, or you can throw it away. So the NPS is macronutrient. This is a composition from the biomolecules such as DNA, RNA, and protein. This is quite simple. That's K, sodium, potassium, sodium, magnesium, and calcium. This is like a single valent cation, divalent cation. They use like for the gradient. They can how to say change the structure of the protein, make a redox of metalloprotein, and also control the osmotic pressure. So somehow they said that we can manipulate the media by the reducing the nitrate component or producing a little bit higher phosphate, and also affected to the plant culture. This is also important. The carbohydrates, this carbon source like a sugar, like a glucose, you can use maltose as well. Plant growth regulator, this is many plants hormone, many plants growth regulators we can choose. Vitamins, this is a cofactor of the biochemical reaction, and some other supplement. Other supplement like the extract, like a casein hydroxide, or you can use the coconut juice. I can see, I saw some colleagues, they use the tomato juice. They use like banana, you see. So we can use the any supplement from the natural and put it in the media, and but sterilize before introducing the plant enzyme. Right. This is a plant growth regulator. It's quite important. They have auxin that many times, a synthetic one. This is 2,4-D, IBA, and IAA. The IAA is a little bit sensitive to the heat. That's we need to filter to the membrane. And cytokinin, that's a synthetic one, VA, and kinetin. This is a synthetic one, but CD18 is the original. So as a heat sensitive, also gibberellic acid. This is an example. This is the structure of the oxy, right? This is a tryptophan-based structure. This one, yeah. So this is a synthetic. So you can choose and try and see the effect of that. Yes. The signaling pathway that I would like to focus, because of the gene expression, this depends on the trigger, auxin, light, ethylene, salicylic, in everything to make a defense mechanism, right? I think I'm a little bit after time. So I will go a little bit faster. This is class induction. That's choosing the explant. This is the study. They use the different kind of the explant to say leaf petiole, leaf base, leaf lamina, and it produce different percentage of the colors production. So the producing the explant, we use the many types of the sterilizing agent, but the common one they use sodium hypochlorite. So I'm go on now. So the light quality also affected growth of the tissue culture. I will a little bit go faster. This is the growth measurement you can study. And I would like to focus the last part of the secondary metabolite production. That's the growth curve. Normally, when the metabolite is producing the compounds in the initial phase, we call primary metabolite, like a tryptophan, like a tryptamine, like geraniol, something like that. And secondary metabolite producing for the defense mechanism a little bit later of the growth phase. So in a secondary metabolite, so you have to study which kind of the production in the cell. This is one example that I reported many years ago about geraniol in the Croton stellatopilosus. They found that one media composed of the BD22 here. This is the code of the plant growth regulator manipulation, and then we found a high yield of the GGOS in the lag phase. So I was discussed with the professor, and then the professor said, this is a primary metabolite. This is true because the GGOH will be substrate of the final product of this plant. So the study of the textures is another thing. This is a packet texture. I will go and we'll finish for in 5 minutes. This is a packet texture. We have to think about the biosynthesis. Actually, we study about the plant tissue culture. We have to think about the plant biosynthesis every time I do that. So we study on that, and we try to manipulate to see the structure, what kind of the supplement, what kind of the trigger, what kind of the elicitor can put in the media to enhance the production. So the first manipulation, we change, not we, that is they change the composition of the media, and they found the pachytextile higher production. They have the two-state culture. One state is for growth production, a second state for the texane or pachytextile production. So we have to manipulate the composition. Once we manipulate the composition, we can select the high yield because every cell cannot produce the similar amount of the secondary metabolites. Once you find the right one, high yielding cell line, you got the good quality of the cell line. And then you control that cell line for further production. This is improve the production of the secondary metabolite. Maybe you can addition of the precursor. I have one papers that I show that limitation of tryptophan that in the cell culture limit the production of acute culture, that's limit production on the methionine in cotton plant. Once I supply the tryptophan, I supply tryptamine, the enhancing of the methionine is also occur. So in the same idea that I got from the pachytextile, we add a precursor, they add there's some vitamins, they add some antioxidant inside, and they produce the higher yield of the pachytextile. This is another method that is two-phase culture. This is key that you can see the concept of the two-phase culture. Because of the plant cell, they have a vacuole, but they can accumulate in the limit portion. If you find out the artificial, like a liquid, which miss immiscible to the media, but they also not destroy your compound. Once the plant cell producing and they release to that liquid, and then they still, oh, no feedback inhibition, I can grow further, I can produce further, and it will enhance. You can learn about the plumbagen biosynthesis. They use the root culture of the plumbago indica. They produce, I think, more than 20 times of the plumbagen production comparing to the one phase. This is two phase, right? Okay. Elicitation, this is also interesting. And elicitation, that's very close to how to say molecular biology. You can use elicitation, whatever you want. They divide the biotic. This is a biotic origin. The abiotic is like a physical mechanism of that. And then the genes in the cell will trigger a different mechanism, producing, enhancing transcription level, enzyme production, and then they produce higher yield of the secondary metabolite. At that period, we will call phytoalexin. This is, you can see the concept in the textbook. So this is the study. What I would like to focus is that if you choose like a chitosan in this way, chitosan in specific concentration, they enhance the amount of the artemisinin, also the period of time. So study of elicitor, we need to study concentration of the elicitor, which one is appropriate. And the second is time, contact, the period of time, which appropriate to enhance. So for the case of chitosan, it means that longer is better, higher concentration is better. But I would say in the methyl jasmonate here, only 4 days, and in a higher concentration. So it means that you have to find the appropriate concentration, appropriate time of the contact cell to the elicitor. This is a periclitic cell that shows that some elicitor enhance the production. Yes. So we can put all methodology like additional of the precursor and elicitation all together. And then will be, if the cell can produce, not going to die, and then you can have a high yield of the production. I have tried one with the kratom, but kratom not survive after pull method after that. So there's immobilization, but this immobilization, I think the application not so high. No? Organ culture, this is another thing. I am not successful with the kratom of the callus culture and suspension culture to produce methionine. But once I produce organ culture like a shoot culture, I got the methionine. So meaning that they have a specific organ to accumulate it. In case of plumbagen as well, there's a plumbagen, they accumulate in the root. Normally in the plant, they accumulate plumbagen in the root. So growing the callus, they cannot. So you can see here the picture. This is from my colleague. She is in Bangkok, and then she got a PhD from this, and then she shared me this slide, and I found it's very interesting. This is callus culture. You see, this is really nice, right? Once you have an organ culture, like a root culture, they have the plumbagen, and also produce metabolites. And from this metabolite, she found that this plumbagic acid and then glucoside is a precursor for the plumbagen. So this is, you see, now plant tissue culture put myself a little bit more, seeing the plant is not producing secondary metabolite only, but also like a factory that we can manipulate what we want, right? So this is hairy root culture. I told you that's an induction with agrobacterium transform hairy root. This is quite interesting. We found that only a leaf vein produce a good induction, a hairy root induction. By this hairy root characteristic that I found here, this is a longer root compared to the root producing from the stem. So we use this and collect it and growing in the media, and then we get the root, hairy root culture. But my hairy root culture only accumulate triterpenoids, not methionine or any alkaloid, right? So it was a little bit disappointed, but finding out what a triterpenoid is not so bad, because triterpenoids in kratom also playing in anti-inflammatory activity. Yes. This is another study about the hair root culture. This is a supplement with the elicitor, right? So faster. This is a micropropagation. They have steps. There's establishment, multiplication, many shoots, and then from shoot to the root, and from the root to acclimatization. I think I reported about the micropropagation. I'm not sure in that one, but the one plant growth hormone is quite interesting in my lab is a thidazolam. It is a cytokinin-like substance that I supply to the median. I got a lot of shoot this here. This is, you see, playing with the ratio of the BA kinetin to the thidazolam. And we got different morphology of the shoot. So we choose the right concentration of TDSAT and the right concentration of BA to produce a shoot now in our lab. And the root, for the root, once you just put the plant to the non-supplement any hormone, root also produce, but a little bit shorter time, I mean longer time. When you use the NAA, you have different, right, root from the IBA, so you can study. But interesting, the finding of 2,4-D, we got a lot of the colors up here. That's why we got the colors. After that, we use the 2,4-D for initiate the colors for kratom plant. Yes. The root formation, that's cutting root to the media, rooting media, or cutting and putting in the soilless or artificial soil, and you get a complete plant that you can grow in nature. This is shoot induction media. This is kratom now, or kratom. This is a shoot induction media. You can see no root here, but a lot of shoot. But here, you see, this is the raised strain of the methagenize basilosa. Once we move to the root induction media, there's no rate at all. I discussed with the professor. He said that maybe because of the thidazolam, they produce the rate. I agree with him. Yes. And then from the root and the shoot and produce here in the bigger size, and then we put in the soil and acclimatization state. I show here. So this is the plant, and move to the vermiculite. And from that vermiculite, we adjust the moisture like a little bit to gradually attach to the normal environment, and then growing in the pot, and we get the plant. So this is you see the uniformity of the plant, as you can see after micropropagation. Yes. So I think, 10 minutes, I'm sorry. So I think in plant culture now, I can inspire all of you to study and initiate the plant culture, which kind of plant, whatsoever. So you can study about that. Yes. Okay. Thank you for all your attention.
Okay. Thank you for your insightful presentation, Prof. Juraiti.
I'm sorry, a little bit fast. So
No problem. Okay. Next,
well,
we
if you
will
have
move
a question.
on the Q&A session. For participants who would like to ask questions directly, please click the raise hand button. Then state your full name followed by your question. As for the participant who would like to ask question indirectly, please type your question in the chat column. Prof, we already have several questions in the chat column.
Yeah, Professor Sam, so do you have a question?
Nothing is.
Yeah. Thank you uh, for your excellent presentation, Dr. Juraitip. We don't have tissue culture. Maybe we next plan tissue culture. And I ask you about which explain is better for kraton tissue culture, leaves, seed, or roots? Which better? Yeah. And second, is it possible for our student to take tissue culture course in your lab? And what are the requirements? Thank you. Yes.
For the second question, I cannot answer you now. I have to ask the academic service about that.
Okay.
And first question, I think after, let's say, 17 years study about the kraton tissue culture, I would say that undifferentiated cell like a color and suspension, they cannot produce a good yield of the mitokinine because of the organelles organization, in the cell. I would say it's quite related to the chloroplast. So it means that now we have the shoot culture and the plant culture. A plant culture means that the whole plant that you can grow in a controlled condition, you can put the plant, because I'm writing the paper about the effect of the lights. There's a blue light and red lights. Yeah, we have finished that one. And we found that the ratio of the lights also triggering the production of the mitokinine. So producing to make plant cell tissue culture to produce mitokinine, at the moment we succeed by the shoot culture and the plant culture. Yes.
Okay.
But if please interesting, maybe using color cell and then try to, how to say, manipulate it in many ways, he can try. And but I would say that in kraton plant, it is not only mitokinine, but also a lot of the flavonoids there. That flavonoids also perform a good bioactivity. So now we focus not only alkaloid, but non-alkaloid as well. We're interested in the tritropenoids, we're interested in the flavonoid. And one colleague took my culture to screen. They found that they said that the HPLC found the flavonoids. The flavonoids is interesting, the name of the flavonoids. Yeah. So that's why the kraton is very good in antioxidant. Yes. Mitokinine is the compound interesting, yeah. But mitokinine also, they have another dark side, right? This is a narcotic one. But now working with the plant tissue culture, I would say, just say non-narcotic kraton, right? Yeah, you get my idea, right? Yeah.
Okay, thank you, Prof. Mr. Januarto, please.
Okay, thank you, moderator, Maspri. Good morning, Prof. Juraitip. Thank you for your presentation, which is very insightful and then very informative. Actually, currently, I'm also planning to learn more about the plant tissue culture for my doctoral program. And I have several questions from your presentation before. Just correct me if I get it wrong. So from what I get from your presentation before is the plant tissue. The principle is we can produce the callus, which is the undifferentiated cell, and then this callus can be differentiated, and then will be full. The will be a full plantlet. When we produce the callus, we can modify anything that we want. Which is maybe I combine with the elicitation method that you also mentioned before with the phytoalexin concept. Now, my question is, when we do some modification in the callus, into the media and then they are going to into the planet, is it that these changes is permanently or just only the effect is just temporarily because there's only the coping mechanism of the stress that we build into the callus formation itself. So when we are transfer into the acclimatization and then we transfer into the soil, is it the plant that we produce from the tissue culture will be have the same characteristic with in the media that we are produced in the lab? That's my question. Thank you, Prof.
Yeah, this elicitation normally is a transient production. A transient, I mean a non-permanently. Yeah. So if we would like to produce a higher yields, we put elicitation like Matthew just monate, maybe just only 12 hours or 24 hours we harvest it. Right? So it's elicitation. You're right that it is not permanent, that if you put elicitation, the plant grow, produce a higher yield of the secondary metabolite. From that plant, move to the soil. They do not remember, they forget already. So if you would like to still, how to say, if you would like to transfer to the plant, you have to work with the molecular biology, with the transgenic plant. Like you have a callus, you have a gene of interest that's produce a higher amount of secondary metabolite. You introduce the gene to the cell, and then from that cell, you produce the plant, and you select the plant that contain the genes, and then you can grow at any time and produce constantly. So by means of elicitation, only transient expression of the genes, transient production of the secondary metabolite. So in case of elicitation, I would say like if you have a plant and you say that, okay, I spray with methylgesmonate, after 24 hours you harvest it, you have a high amount of the secondary metabolite. You understand what I explained, right? Yes, the concept.
Yes, yes.
Mm-hmm.
Yes. So it means if we want to produce the permanent plantlet, you said we need to work with the molecular biology.
Right.
Now, the follow-up question with that, is it the plantlet when we working with the molecular biology is considered as the GMO, like the genetic modified
It is
organism or
true.
not,
It is true. It is a
Professor?
GMO, but this GMO is for medicinal purpose. You don't eat directly. You have to extract it, right? If you have a GMO potato, if you have GMO tomato, you eat directly. You have the viron, we have a vector. But the medicinal plant, you make a GMO. If you would like to have the compounds, you have to isolate it. It means that the vectors are gone, the contaminants of the GMO process are gone. You get only the metabolites. This is different between medicinal plant and food. Okay.
Okay, noted. Thank you, Prof.
Okay, thank you, Januarto.
Yeah, thank you,
Okay,
Prof.
next move the chat box, yes, from Maria Melda Mola.
What are common contamination issue in the medicinal plant tissue culture and how can they prevent it? A common contamination is fungus. A lot of fungus contamination. So we have to use the right appropriate sterilization method to produce it. Yeah, I got up fungus, many types of the fungus. Yeah. We have to use many types of the sterilization method.
Okay, thank you, Prof. Next, the raise hand. Prof. Sam, raise hand. Oh, okay. Prof, I'm question myself terkait apa namanya study research kratom. How efficient in the genetic transformation of Agrobacterium rhizogenes in Mitragina speciosa, and to what extent does the application of elicitor and biosynthetic precursor enhance of the production of nitrogen and secondary metabolite compared to mature plant tissue?
You can learn from the papers that I published in 2007 to 007, I think, to 008 with the grow bacteria of producing the grow bacterium and then infect to the plant and then growing only the root. And from that root, I did not use any elicitor. So if you're interested in, you can produce the one, right? And then working with elicitors. But normally the accumulation site of the metagenin is not in the root. That's in the root, maybe you can find, if you're successful, you can find the ox-indo-alkaloid. You know the word ox-indo-alkaloid. This is another type of the compounds accumulated in a kratom plant, and it show a good, bioactivity as well. So if you're not remitted to the metagenin only, you can also find another type of the alkaloids in the root. It's interesting. You can do it.
Okay, thank you, Prof. And next, the chat box from the Lintang Cahyani.
What is the key challenge in establishing a successful medicinal plant tissue culture protocol? The key success is you work hard. You have to work very hard and very patient because the plant grows so slow. And you have to be, how to say, a good design of the laboratory, experiment, because once you get, how to say, you have to decide very precisely. You produce a bunch of the plant and you manipulated a bunch of the plant and you got the result. So you have to spare of the plant because of the first experiment, maybe you have to adjust something and you have to go back. If the plant grow 2 months for the cycle, it means that your time is a little late for 2 months. So I think the key challenge would be a good design of the experiment, patient, and work hard. Yes.
Thank you. Okay. Next, participant. Okay.
But I would say application now, the plant tissue culture, they use like expression protein. Now we very familiar to the vaccine, right? To the vaccine, like a COVID vaccine, the vaccine for the anti-cancer in many cases. They are also interesting to produce in the plant cell. You can see in the reviews in the online textbook, and then you can study about that. And now they focus not only produce the medicinal plant, but they also use the plant tissue culture as a factory of the producing a protein, a therapeutic protein, I would say, therapeutic proteins. This is another way to do with the plant tissue culture. I would say plant tissue culture is like in the middle way that you can put the molecular biology there, you can put everything, and then you get the result, protein metabolized in everything that you can plan.
Okay, thank you, Prof. Okay, thank you for your answer and to all participants. And the maximum time for the Q&A session has been reached. Declaration that Q&A session is over. Okay,
Thank you.
let me summarize the material from this topic. Meristematic tissue play an important role in plant regeneration because it has the ability to reproduce and generate various new plant parts. Plant tissue culture utilizes part of plant with regenerative capability such as meristem to produce plant more quickly and efficiently. One of the main techniques used in tissue culture is callus culture, where plant tissue culture induced to grow from cut or damaged section can develop into new plant. This technique is useful for developing plant in culture media that containing specific nutrient that support faster and more abundant callus formation. Additionally, cell suspension culture allow plant cell to grow in liquid, which is used for producing bioactive compound like taxol, as well for further research into factor the affect cell growth and secondary metabolic production. One of the key areas of research in tissue culture, the optimization of culture condition, which involves adjusting various physical and chemical factors in the culture medium. Factors use pH, temperature, and media composite are crucial in supporting efficient plant growth and production. Moreover, research focus and selection cell line that can withstand extreme conditions, whether in terms of terminal temperature of chemical composition in the culture medium. The cell can grow better even under less than ideal environmental pressure, making them prime candidate for industrial application and future research. High root culture is very useful technique in accelerating the regeneration process, particularly for producing root that develop faster in large number. This technique has significant advance in setting up the plant life cycle and regeneration, as will production root that can be used for secondary metabolic production or further regeneration. Additionally, organ culture, which involve culturing part of plants such as root, stem, or leaves, is also used for plant regeneration. This technique allows the formation of new shoot or rooting using specific growth regulation such as TDZ, which stimulation the formation or more shoot. Root development is also integral part of this technique where external factors like chemical solution concentration of temperature influence root development for stronger plant. Overall, plant tissue culture offers significant benefit in efficiently production superior plant, accelerating regeneration and generating bioactive compound with high economic value. By optimizing this technique, better result can achieve in modern agriculture, biotechnology, and the pharmaceutical industry. Okay, close. And the end Q&A session mark the end of this lecture. I hope we have all participants would like to thank you, Prof. Juraitif, for this willingness to deliver this knowledge on the role medicinal plant tissue culture. And on behalf the committee would also like to thank all participants who were already active during the question session. So, Prof. Juraitif, the committee has something to present to you, appreciation for your effort. Please accept the present certificate. Committee certificate. please. Yes.
Thank you very much. You can send this picture to my email.
Yes.
Thank
Okay.
you.
Thank you. Thank you, Prof.
Yeah.
Okay. That's all for today presentation. Once again, thanks to you. Good afternoon and best wish. Next, return this event of the Master of Ceremony. Thank you.
All right, thank you so much, Mr. Kristiantoro, who will lead this session properly. And also, thank you so much for Dr. Rana Juraitip. Your knowledge and insight have truly inspired us today, and we are grateful for the time and effort that you dedicated to the session. And also, like the emphasizing by our dean that as we know that we not yet have a tissue culture laboratory. So maybe we can have a further collaboration with Songkla University. So maybe we can conduct our research there.
Yeah.
All right. Thank you so much. Once again, I say thank you, deepest gratitude to our esteemed speaker for sharing your valuable expertise on medicinal and plant tissue culture. Thank you so much.
My pleasure. Thank you. Yeah. All right.
All right. Special thanks also to our wonderful moderator, Mr. Kristiantoro, Magister Pharmacy, for skillfully guiding this discussion and ensuring a smooth and engaging session. And for that, please accept our certificate of appreciation to our wonderful moderator. Kamita, please share.
Thank you. Thank you, Amalia.
All right. Thank you so much for your contribution, and your contribution have greatly enriched today's experiences. Thank you. All right. Ladies and gentlemen, as we conclude today's an insightful discussion with us, and of course, I want to thanks to all the participants that your enthusiasm and thoughtful questions and also your engagement made this even more meaningful. On behalf of Faculty of Pharmacy, University of Pancasila, we truly appreciate your presence and participations. We look forward to seeing you in our future events. By that, it's all the end of our event today. Have a great day. Wassalamualaikum warahmatullahi wabarakatuh. Shalom, Om Santi Santi Om, Namo Buddhaya, Salam Pancasila.
So adorable. Thank you, Amalia.
Thank you, Doctor.
Yes.
Thank you, Prof.
Okay.
Juraitip.
Yeah. See you later then.
Yes, thank you.
Okay. Bye-bye for all. Thank you. I'm leaving. Yeah.
Thank you so much.